So Yangning Liu is a postdoc at MIT and Howard Hughes Medical Institute. And during his doctoral studies, he worked with C. elegans neurocircuitry and showed the importance of extrasynaptic transmission and neural multitasking in circuit function. That's what he will be showing us today. And now he is a postdoc. He is developing novel tools that will facilitate the study of neurocircuits in general. So the floor is yours, Yangning.
Thank you, Mengsen, for the nice introduction, and really appreciate this very nice conference to have me. So what I'm going to talk about today is how symmetric behavior is generated from asymmetric wiring. So in most animals, particularly bilaterans, there are distinct locomotion forms, mostly composed of undulation, or a mixture of undulation plus limb movements, or simply limb movements. And one of the key hallmarks of these behaviors is the left-right symmetry. Underlying the left-right symmetry is the left-right symmetry of the neurons that control this behavior. Take, for example, in the Leftright locomotor center, there are excitatory motor neurons that activate left and right sides equally. In between them, there are commissure neurons that relay the cross inhibition. So that these two left-right locomotor centers can coordinate antagonizing each other and at the same time generate an equal level of locomotor behaviors on left and right sides. In the first larval stage of C. elegans, which is the youngest larval stage of C. elegans, it's generating this very balanced sinusoidal movement on its dorsal and ventral sides. So in C. elegans, the animal crawls on its lateral sides. And by generating bends on dorsal and ventral muscles. So basically dorsal and ventral sides in C. elegans can be considered as left and right in most bilaterans. This is not very surprising and very normal. However, if you look at its locomotor center, it's very surprising that it's composed of a highly polarized dorsal ventral wiring system. Specifically, it's composed of excitatory premotor interneurons that synapse onto excitatory motor neurons, which is acetylcholine, a releasing neuron, that only activates synapses onto the dorsal muscle. At the same time, it innervates the GABAergic neuron that only innervates the ventral muscle. So basically, with this wiring diagram, you don't expect the behavior to be dorsal and ventral symmetric at all. You expect the dorsal muscle to be contracting all the time, whereas the ventral muscle relaxes all the time. However, as you just saw, this is absolutely not the case. So curious about how the muscles actually behave during the L1, the first larval stage C. elegans locomotion. We used the genetically encoded calcium indicator GCAMP and expressed it in the muscles with an RFP as a ratiometric indicator. The ratio of these two would tell us how the muscles change its activity during the locomotion. So we developed segmentation algorithms to divide the animal into all these segments, and then we can accurately quantify how the activity is behaving during the locomotion. So remember, the motor circuit that I just mentioned only controls the posterior body wall muscle, which is the 65% of its body on the posterior side. The head muscle is controlled by a totally different system, which is symmetric. And when we quantify the muscle activity propagations, we can see clearly that the dorsal muscle, as well as the ventral muscles, they output roughly equal amount of total activity for the body wall muscles. It's only the pattern that overlaps that antagonize each other. The overall level of output is completely equal. And that's consistent with what we saw from our naked eyes, that yes, it's producing a sinusoidal movement with no bias. So the question becomes, how does a structurally asymmetric circuit generate symmetric locomotor output? So to answer this question, people in my previous lab developed high throughput electron microscopy reconstruction strategies, and they actually identified additional cells that were previously uncharacterized from the first EM study done in 1978. They actually identified additional excitatory neurons that innervate dorsal and ventral muscles, as well as another cell called HMC, which is not a neuron, but also has gap junctions with dorsal and ventral muscles. So together, this is the wiring diagram we have, but still it doesn't look very symmetric either. And we want to address how the symmetric behavior activity comes from. Over the past 40 years, actually, people have generated various hypotheses regarding this question. For example, many people suggested that the ventral muscle on L1C elegans might be intrinsically active, and it's more has more activity over the dorsal side. Or maybe the ventral muscle is proprioceptive and can be activated by sensing dorsal bending. Or that the GABA released on the ventral side might be excitatory actually to the ventral muscle. Or that the GABAergic motor neurons can co-release unknown excitatory neurotransmitters. Or that there are simply more excitatory motor neurons, which our colleagues discovered from the EM reconstruction. Or that based on the new EM reconstruction, the HMC cell might relay the dorsal muscle activity to the ventral muscles, or maybe some other, which we don't know about. So to tackle all these hypotheses, we established two scenarios where we first established a correlation between these cell activities with respect to the locomotion. And second, we established the causal relationship between these cells' activities and muscle activities. For the first approach, we simply used calcium imaging during free moving animals. And take, for example, where in this particular animal, we label one type of motor neurons, and this type of motor neurons are activating only during the backward locomotion, which is highlighted in the right panels. And because of their anatomical wirings with respect to the body curvature change, we can establish that this particular type of cells correlate with dorsal bending during backward locomotions. So this is the correlation that, strategy that we used. For the causal relationship establishment, we used the simultaneous manipulation and monitor of the circuits. So to do that, we used optogenetics, which is widely used nowadays, and couple it with calcium imaging. One of the well-known problems with this optical interrogation approaches is that there are spectral overlaps between opsins as well as the G-camps, for example. That means that when you turn on the light, you're already activating the cells, and you have no reference as to how the G-camp looks like before the activation takes place. So people have largely used two approaches to address this problem. One is to use spectrally shifted opsins and couple it with G-camps, for example. And another is to use spatially pattern light to basically specifically target cells of interest, and then without touching cells on outside of the cells of interest. However, these two approaches are both requiring technical challenges, and we probably want to make new animals if you want to do that. To take advantage of the fact that the G-camp actually has a delay with respect to the onset of the cell activity, we reasoned that we don't need anything complicated like that. So imagine how the cell looks like pre-stimulation, during stimulation, post-stimulation. Ideally, it looks like this. What the G-camp will look like will be something like this, the red line. That means by the time you turn on the cell, you activate the cell, the G-camp doesn't have enough time to reach its peak value. It takes about a few hundred milliseconds to reach its full peak value. And therefore, by only recording and illuminating at the same time, activating the opsin at the same time, the first data point you acquired is reflecting what happened before stimulation. And at the end of the stimulation, you're going to see what the cell looks like after the stimulation. And therefore, we only need to record and illuminate the opsons at the same time. And by capturing the first few seconds or a few hundred milliseconds, you have a reference point already. And that approach is very inclusive. That approach doesn't care which particular opson, which particular wavelengths you use at all. So using this approach, we basically spent 3 years and concluded nicely that none of these hypotheses are valid. And it's including the fact that the muscles may be autonomously active. We also excluded this hypothesis either. Okay, so this is very unfortunate. However, there's one thing that's left, which we never touched, which is the excitatory premotor interneurons. Okay, this type of premotor interneurons, in C. elegans, there are only 5 of them. And 2 of them control the forward locomotion, 3 of them control the backward locomotion. And interestingly, all these premotor interneurons have this long process that lies along the ventral nerve cord. It's actually in proximity towards the ventral muscle cells. And all of them are cholinergic. All right, so this actually set up the realm of our hypothesis that actually maybe, just the maybe, that these premotor interneurons might be underlying the ventral muscle excitability. So to test this hypothesis, we used the optical interrogation strategy, which we described, and expressed general opsin in one of those premotor interneurons. And to avoid the GABA synapse releasing that might be complicating our results, we used a mutant that doesn't release GABA, that doesn't synthesize GABA, so that we can actually purely see what would happen when we activate the premotor interneurons. So to our surprise, yes, it's actually activating the ventral muscles by simply activating the premotor interneurons. And as you can see on the ventral side, the activities are increased upon activation of one of those premotor interneurons. And this is highly satisfying, that this is the first time we can actually get a neuron that's activating only the ventral side. And on contrary, when we optogenetically silenced the premotor interneurons altogether and recruited the muscle cells, we saw that, yes, the ventral, as well as actually the dorsal cell muscles, are inhibited. So this is sufficiency and necessity. However, both of these approaches are optical interrogations. And we wanted to validate our results by other approaches. So we simply used a light-sensitive protein that when activated can ablate cells where it's expressed. And using this, after we ablate all the premotor interneurons, what we saw is actually very satisfying, where that we can only see the remaining of the dorsal activity, but the ventral muscle activity is pretty much gone. And the fact that we can see the dorsal activity is consistent with our previous findings, where upon ablation of premotor interneurons, the excitatory motor neurons are central pattern generators, where they can generate activities that activates the dorsal side. So this is consistent with our previous finding for the central pattern generators. What's really unexpected is that we saw a loss of ventral muscle activity in this approach. And when we saw
Yangning, you have to wrap up now.
Okay, very fast. So when you look at the behavior, you can see that the dorsal side is always coiled. So in conclusion, so additional experiment where we spread out the sort of coiling, we actually saw the muscle activity being expanded only on the ventral side. And this is highly satisfying as well. So in conclusion, we found that the premotor interneurons have dual roles, that it's not only the premotor interneurons, actually motor neuron by themselves, and that extracellular transmission compensates this structural asymmetry. And by additional stories, we also found that the GABA is also acting upon the excitatory motor neurons, so that we have a balanced outputs on both sides. In conclusion, we have basically identified the extracellular transmission that was previously unknown from the premotor interneurons that activates the ventral muscle cells in L1C elegans, and that by negative feedback, you can form a highly symmetric but structurally complementary shape of dorsal and ventral muscle cells activity patterns, which is what you can actually achieve by asymmetric wiring, but they take advantage of the asymmetry here. So with that, I'd like to thank all the lab members from my previous PhD study and all the funding sources, collaborators, committees. And with that, I'd like to take questions. Thank you.
Thank you so much for the amazing talk. Let's see if there's any questions. This session, encourage people to ask questions. For me, this is quite amazing. I was wondering if I guess you're still in the stage. This field is still in the stage of figuring out what are the neurons or how they're connected. I was thinking I'm a computational neuroscientist, so I think more in terms of if anyone has done the modeling of this stuff. Even with very simple neurons, their behavior could be quite complicated, and they will be able to generate rhythmic pattern despite there may not be very complicated wiring. So what are the major perspective on that?
Yeah, it's a very nice question. Yeah, so actually, we are trying to model how the system works, and but of course there are a lot of parameters which we would have to fit to actually get the results similar to what we saw. But yes, there are efforts currently being done to try to simulate this the entire system. And it looks like this system is more complicated than we thought. But the fact that it's actually using fewer, only about half of the neurons where the adult C. elegans uses, it's still able to generate similar behavior. And this is really inspiring to us because it looks like it is possible that even with a loss as much as half of the neurons, it is still possible for you to converge onto the behavior that that's also implying that the loss of motor neurons in some of those diseases, neurodegenerative diseases, those models, it is possible for those systems to adapt similar strategies to achieve functional outputs at the similar to the wild type. Yeah, but like your question is, yes, the efforts are being done, but we haven't actually finished this non-trivial task. Which
looking forward to any upcoming studies.
Yes.
So it looks like we're at the end of this session, and I want to thank all the speakers, and I want to thank myself for asking questions, and thank you so much for presenting amazing work. I hope you enjoy the rest of the conference, and I hope to see you around at the conference. And we're going to end here. Have a good one.
Thank you. Take care.